The broadcast is now starting. All attendees are in listen-only mode.
Hi, thank you for joining us today. We have Luke Flory. He'll be giving a presentation on plant invasions. My name is Rebecca Wallace. I work at the University of Georgia, and I'll be kind of moderating this a little bit. This will be made available. We are recording it, and it will be made available through potentially the NAIPC website. I believe it may also be made available through the Bugwood YouTube account. So if anybody has any questions about that, they can email us to, get a link to that as needed. And I will be you can enter your questions throughout the presentation in the chat box. And at the end of the presentation, I'll read off the questions to Dr. Flory, and he'll answer them. That way they can make their way very easily into the recording. So without any further ado, Dr. Flory.
Thank you, Rebecca, and thank you all for joining us today. I'll provide a brief introduction to what I do and a little bit of my background on what we do here in my lab at University of Florida. I grew up in Northwest Indiana on a small farm and went to DePauw University, a small private liberal arts school, for a biology degree before doing graduate work at Indiana University. And I have a long-standing interest in non-native plant invasions. I have a degree in applied ecology and a PhD in ecology and evolution, and have worked on plant invasions for more than 15 years, nearly 20 years now if I count the intern work I did as an undergrad. In my lab here at the University of Florida, where I've been for almost 8 years now, we mostly focus on basic questions about the ecology and impacts and mechanisms of plant invasions, but we also try to answer questions that have management implications or relevance to management in some way. So today, what I want to do is to do two things: provide a broad overview of the types of things we do in our lab by working through a number of different study systems and types of questions that we ask. But I also want to try to pull these different projects together into a broad framework of understanding drivers of long-term dynamics and impacts of plant invasions. Okay, so what you're looking at here in this photo is a view from the top of the old Los Picachos volcano on the island of Santa Cruz in the Galapagos. And of course, the Galapagos are an incredibly important biodiversity resource, many endemic species, many threatened species as well. But what you're also looking at in this photo is a number of non-native and invasive species, which threaten many parts of the Galapagos. And one species I want to point out in particular as an example of the types of things I want to talk about today is Cinchona pubescens, or quinine tree. This species was introduced into the Galapagos decades ago because of the anti-malarial properties of quinine. It was introduced into the agricultural zone of Santa Cruz Island. Many people don't know that there's more than 30,000 people that live on the Galapagos, or nearly 30,000, primarily on 4 different islands, and that 2 of those islands have agricultural zones. On Santa Cruz Island, Cinchona escaped the agricultural zone and invaded a wide range of areas. You see a photo here from the 1990s where it has spread around this Los Picachos volcano. Well, when I visited the Galapagos last year, it looked like this. And so what you see here are skeletons of these adult Cinchona trees, and these trees have not been managed in any way. They've actually succumbed naturally to what we now believe is the pathogen Phytophthora cinnamomi, which is in itself one of the most invasive pathogens worldwide. And in collaboration with Heinke Jager at the Charles Darwin Foundation and one of my former PhD students, Kathy Fahey, we're working out the population biology of Cinchona and then how it's altering effects on native species, the lack of the invasion at this point, altering the response of native species. And so I want to highlight this, and I'm not going to show you any of the data from this project right now, but I want to highlight this as an example of where we really needed to understand the possibility in terms of what would happen to this species over the long term. And so we couldn't predict that this pathogen would, as you might say, catch up with this invader, but if we could predict it, we may allocate management resources more effectively. But before I start talking about specific projects, I want to take a step back just for a moment to remind us how we got to this point. And so, of course, we know that we've been in a period of exponential population growth since at least the 1960s. At this point, we have 7.7 billion people on Earth. We're projected to have 9 billion people by 2050. And of course, given the increasing population of the Earth, acceleration of technology, we're moving around the world like never before. And so if we think about global flight patterns and the movement of people and where these people might be going, a lot of this is for tourism. And if you've never seen the site FlightAware, I'd encourage you to check it out. They report that at any moment, there's an average of 9,000 planes in the air that are carrying 1.3 million people. And many of those people are visiting natural areas, such as these people visiting the California super bloom earlier this year. And maybe you saw the news report that somebody even landed a private helicopter on a nature reserve. And at the same time, we're moving goods around the world like never before, with shipping containers and large ships, among other methods. And the World Shipping Council reports that we're losing 1,400 of these containers a year. And at the same time, in addition to accidental introductions due to global travel and trade, there's also the intentional movement of species for horticulture and agriculture and other purposes. And so at this point, we have more than 13,000 naturalized vascular plant species around the world. This is a really nice paper by Mark Van Kloonen's group and others in 2015 in Nature, showing both the routes of introduction, which regions contributed and which regions received these non-native species, and then the hotspots of where these species are concentrated. We have more than 1,800 in Florida alone. And of course, only a subset of these non-native species are invasive, but those that are, of course, can cause major impacts on biodiversity and ecosystem functions. At the same time, there's no indication that the rate of introduction of species is slowing down. And so in this paper, they reported that for many taxonomic groups, the first record of non-native species in a new range, including plants and birds, amphibians, and insects, continues to increase. What we are getting a really good handle on, or pretty good handle on at this point, is the impacts that invaders are having. And so over the last 5 or 10 years, we've had a number of really nice meta-analyses and syntheses and reviews documenting the ecological and sometimes the economic impacts of plant invaders. However, many of these invasion impact studies are very short-term. And so this is a review paper from my research group that we put out a couple of years ago. And so we documented the number of papers on the ecological effects of plant invasions, and we show an exponential increase over time. However, most of these studies are very short duration. So whether we look at observational, experimental removal, or experimental addition studies, most studies are conducted for less than 1 year, and a few studies are conducted for 1 or 2 years, but only 8% of studies have been conducted over 4 years or more. And at the same time, we're spending more and more money on managing these plant invaders. And so this is a paper that'll be coming out in the next few weeks from my lab group. We've been working on this analysis for a number of years. It took a long time to gather the data and analyze it. Here we're reporting how much it's costing us in the state of Florida to manage invaders in natural areas alone. So this doesn't include private lands management or the county level management, but we're looking at $45 million in cost per year for a number of different invaders, primarily focused on about 10 invaders, with hydrilla being our most costly invader. And so this leads to this overarching question of what are these mechanisms, what are these drivers in terms of biotic and abiotic factors that determine long-term patterns in plant invasions, dynamics, and impacts. And this is a basic ecological question of interest. But in addition, it has management implication in that we have to allocate limited funds towards invaders, preferably towards those that are the most problematic and that are the most persistent. So what I've done is to pull together some of the possible drivers of long-term patterns. And many of these ideas come from David Strayer's 2006 paper in Trends in Ecology and Evolution, where he was thinking about some of these same ideas. And so what I'm showing here are some possible biotic drivers, including phenotypic plasticity and evolution, which we would both think would have positive effects on native and invasive species during the invasion process. So For example, a native species that's more phenotypically plastic may be better able to tolerate an invader. An invader may evolve to be more invasive over time. However, for either the release or the accumulation of enemies, including insects or pathogens, or changes in community composition, including successional processes, we have there could be multiple hypotheses in terms of direction of change for native and invasive species. For abiotic factors, including physical structure, nutrient availability, hydrology, and fire, generally, if those changes, those abiotic changes, are due to the effects of the invader, we would expect a negative effect on the native species and a positive effect on the invader. Otherwise, the invader would not be an invasive species. You will notice the asterisk there at the bottom that these changes in physical structure or nutrient availability may benefit some native species, but as a whole, we would expect a negative effect, I think. For climate change, it's really a mix of possible mechanisms because of the different mechanisms including temperature, drought, rising CO2. There's a lot of different factors, and so the interactions can change in multiple directions. Okay, so before I start talking about these different projects, I want to acknowledge at least some of the people that I've worked with on these. I'm giving this presentation, but of course, this is the result of the work of a wide variety of people, including current, former students and postdocs and collaborators at different universities and organizations. So I'm giving the report here of a lot of work done by multiple people. Okay, so what I'm going to do is focus on two major study systems. I'm going to focus on grass invaders in both cases that I've worked on for much longer than other. But what I want you to do is maybe not think so much about the species in particular, unless that's the species that you're particularly interested in, but try to think about how it relates to your species of interest and if this possible mechanism would hold up for that species you're interested in. So the first species is Microstetium vimineum, or stiltgrass. This is a species that I've worked on for about 15 years now, since I was at Indiana University in South Central Indiana. So this species is an annual. It's native to Eastern Asia. It's a shade-tolerant species, but fairly unusual in that it is a C4 species, and so people have suggested that it's filling an empty niche in the system because most of our understory grasses in the eastern U.S. are C3 species, so it's doing most of its physiological, most of the physiology of the species is at peak during the warm summer months as opposed to the spring and fall like C3 species. And until recently, we've seen very few herbivores and pathogens on the species. And you'll notice from the EDMAPS figure here that the distribution is very broad. At this point, we have it from the Panhandle in Florida all the way north to Detroit, and from eastern Texas all the way to the Atlantic seaboard. So the first study project I want to report on here is this one where we looked at patterns in the phenology of Microstetium across latitudinal gradients, with the idea that the species may have evolved its phenology or its life history patterns based on the growing season length. And so what we did is we collected populations from a wide range, from South Carolina to Connecticut, brought them together into a common garden environment at Rutgers University, and we tracked their patterns in growth in terms of days to anthesis, or the number of days it took for the species to flower. And we found a very striking pattern, whether we exposed the plants to a northern light treatment or a southern light treatment. Plants that were from more northern latitudes flowered much sooner, so fewer days to flowering. Ones in the south took much longer to flower. And to orient you here, the first record of introduction of Microstetium was in Tennessee in 1919. Doesn't mean that's where the species was introduced, just where it was put into an herbarium. But in any case, when we think about those southernmost populations in South Carolina, the first frost is in November, whereas in Connecticut, the first frost is in September, suggesting that the species has evolved to its phenology to match the growing season length. It has to produce seed, flower, and produce seed before the first frost. And so this is important because the longer Microstetium grows before it flowers, of course, the larger it is, and we know the larger the plant is, the more seed it produces. So there's potentially significant implications here for the spread and impact of microstetium. So one example where the long-term dynamics and impacts of this invader could be influenced by evolution of the species. So microstetium can invade a wide range of habitats, from disturbed riparian areas along roads and trails, which is where we often see it first, but it can also just invade shaded, undisturbed forests where there can be less than 5% light. And we have experimental evidence that it can reproduce and produce viable seed at something like 5 to 7% light. And so across these habitats, we see big impacts of the invasion, or we were interested in whether or not we were seeing big impacts of the invasion. And so this photo here is from Big Oaks National Wildlife Refuge in southern Indiana. And this is kind of an open, broken canopy woodland where microstetium does very well. You see this very dense invasion. And so early on, with support from the Nature Conservancy, starting in 2004, 2005, I was interested in what are the impacts here? How can we quantify the impacts of microstetium invasion? And so I set up what eventually became a long-term experimental introduction. And so this is near Bloomington, Indiana, where I set up these 5 by 5 meter plots and in this kind of forest opening you see in the photo at the top. And you can actually see the treatments, the invaded treatments, because microstetium is a C4 grass, senesces earlier in the fall than the C3 dominated native plots. And so all the brown plots you see there in the aerial photo are the invaded plots. In the spring of 2011 and 2012, we divided the plots into 4 quadrats and burned one of them. And so first of all, microstetium did very well in this experiment. And so it was very, very abundant over the first 4 years of the experiment. And we subsequently published a wide range of papers on the effects of microstetium, including how it alters nitrification, how it suppresses native plants and alters diversity, changes soil microbial feedbacks, alters tree survival and growth, and affects insects. However, after the first 4 years, the pattern in microstetium abundance changed quite dramatically. And so what we saw was a population crash of microstetium, whereas by 2012 and 2013, it was essentially gone from those invaded plots. We added that fire in the spring of 2011 and 2012 because we thought there may be a positive feedback of fire, and we temporarily saw a kind of a boost in the population size in those burned areas of the plot. One contributing factor we think for the decline was the severe drought in the Midwest in 2012, but clearly there was a major decline in the population of microstetium well before that 2012 growing season drought. So the change in microstetium dominance over time had significant consequences for change in native species. So here I'm just broadly grouping by functional group, native forbs and graminoids. And for forbs and graminoids early on in the experiment, when microstetium was dominant, there were significant impacts on both functional groups. We then saw some variable impacts once microstetium went into decline and we applied the spring fires. And one notable one is at the top there, where the application of fire resulted in much greater forb biomass in the plots. Another very striking difference that we saw is that species diversity changed dramatically over time. So in 2006, early on in the experiment, even though microstetium was abundant, there was very little difference. There was no difference essentially in native species diversity. Whereas in 2008, there was 40% less diversity in the invaded plots. And by 2013, there was essentially no difference, maybe a little bit higher difference due to the fire treatment. If we look at light availability, which we think is an important resource in the system and maybe driving these differences, at percent light at ground level in the bottom panel, we see very low light availability in the invaded compared to the uninvaded plots in 2006. We see a similar pattern in 2008, but we see no difference in 2013. However, at half a meter, we see low light in the invaded plots compared to the uninvaded, the control plots in 2006. But in 2008, we actually see more light in the invaded than uninvaded plots. We think due to the fact that the invader has suppressed native species that otherwise would be taller and create a more shaded condition. In 2013, we see no difference due to the invasion, but we see much more light availability when we applied fire. So what we think happened here is that without fire, the trees became highly dominant in the plot, there was low light availability, and the invasion declined. When we added fire, tree abundance was reduced, both tree size and density, resulted in high light availability, abundant native forbs, and decline of the invasion. And now I'm not suggesting that light drove the invasion in either case, but the result in terms of succession, whether to tree dominance or forb dominance, the result was that the invasion essentially disappeared from the system. However, we often see that microcesium is very persistent across a wide range of sites. And so we hope clearly in the paper that we're not suggesting that microcesium is just naturally going to go away in most places. So for example, in this site in Virginia, herbarium records suggest that it has been at this site for at least 60 years. And of course, it's a very different site, and so than what we had in our experimental conditions. So we've thought through some of the possible reasons why succession resulted in decline of the invader at our site and not at this site or other sites. So for example, at our site, we had very little disturbance at the site except for those spring fires in 2011 and 2012, which was well after microcesium had gone into decline. And so at other sites, we know, for example, that high abundance or high density of deer may be driving invasions of microcesium, persistence of microcesium. At our site, we also, and at other sites, in terms of succession overwhelming invaders, you would need availability of native propagules. And those propagules, those native species, would need to be highly competitive with the invader. In addition, there needs to be high resource availability. So our experimental site was a bottomland forest site, or would have been, and it had high soil moisture and very rich, high nutrient soils. And then there's this potential role of periodic drought or other climate change factors that may drive a decline of invaders over time. Okay, so now I want to turn to another topic, a biotic driver, another biotic driver of long-term patterns in invasions, and that's the idea of emergence and accumulation of pathogens. And so the idea here is that in the home range of a species, such as the home range of microcesium in China, there are a wide varieties of enemies, either insect enemies, which you see here from photos that I took on a trip back in 2011 to China. You see insect enemies, potentially pathogens, which we know have been documented on microcesium in China, although I don't have any good photos of them. In the home range, the population size, the diversity, or the density and the distribution of the species may be controlled by these enemies. But in the introduced range, such as the invasive range of microcesium in eastern North America, the species escapes those specialist enemies, and any generalist enemies cause relatively little harm. And so we often see microcesium that looks like this, that looks like it's actually been managed for how few pests seem to be on it. And so we don't have good data indicating that microcesium has escaped enemies, but we do think that this mechanism for invasion is likely. However, over time, you may expect that enemies would catch up with an invader, and in fact, that's what we started to document in about 2008, 2009, with the case of this bipolaris foliar fungal pathogen that causes this leaf blight on microcesium. And so we described this disease symptoms that we're seeing, and then we more broadly thought about the role of pathogen accumulation and long-term dynamics of plant invasions. And we gave some examples in this paper, such as the pathogen, the disease that you see on Canada thistle, which you see in panel B there, the mold that you see on garlic mustard in C, or the rust that you see on kudzu in panel F there. And so then we provided this general conceptual idea of how invaders and their pathogens may change in spatial distribution and density over time since introduction. So following the initial introduction of an invader, we hypothesized, of course, that it expands in distribution and density, but then eventually pathogens would catch up and increase in density and distribution, and eventually cause the decline of the invader. And so with the first NSF grant that I had after I arrived here at University of Florida, we sought to explore whether this was happening with microcesium. So the first thing we did was to go out to more than 80 sites in 18 states across the invaded range of microcesium and sample the pathogens that we found on microcesium. And so most of them were from this bipolaris genus, the most widespread of which was the one that you see as represented with the purple circle there, which at the time we called Bipolaris taxon megaspora, but for which we're now in the process of redescribing a historical pathogen called Bipolaris gigantea, which we determined with genetic analysis was actually the same species. And so the pathogen is clearly widely distributed across the invaded range. And so we started to think about possible origins of these pathogens. Were they introduced from the native range, for example? And in GenBank, there's evidence that the same Bipolaris taxon megaspora was found on zoysia grass in Japan, suggesting that introduction from the native range is possible. It's also possible that as the distribution and abundance of microspecium increased, that the invader intersected with native species that hosted these pathogens, such that there was spillover from the native species onto the invader. And so Bipolaris is a common pathogen of grasses, turf grasses, and even corn. And then there also could have been evolutionary changes in the host range of these local pathogens to allow them to infect microspecium. So the next question we had is, are these pathogens actually doing anything when they infect microspecium? So we conducted a simple experiment at one location across 4 sites in West Virginia, where we had plots that were where we applied a water control, or one where we applied fungicide to reduce disease. And we were very effective at reducing disease, and the result in terms of the number of seed heads produced by microspecium was that on average across these 4 sites, seed production was 56% greater in 2012 and 89% greater in 2013, suggesting that this pathogen was in fact suppressing reproduction of microspecium. Now the next question was how the accumulation of pathogens on this invader was affecting interaction with native species. So back with that review paper that we published, we thought about interactions between natives and invaders in the face of pathogen accumulation. So the same axes on this figure here, y-axis is spatial distribution, x-axis is time since introduction. We came up with 3 possible scenarios. So as the invader increases in spatial distribution with time, may cause the decline of the native species. Following pathogen accumulation, which you see in the lightly shaded gray part, panel B, we thought that the first possibility is that the pathogen could accumulate on the invader, and the invader could go into decline, thereby allowing the native species to return. So we called this the pathogen accumulation and invasive decline hypothesis, or PAID. Another possibility, though, is that the accumulation of pathogens may have no effect on the invader, in which case, in terms of the population dynamics, population level effects, in which case the native species would have not be affected either. However, a third really interesting possibility is that the invader could host the pathogen and tolerate the effects of disease, but then the pathogen could spill back onto native species, causing further decline in their population and actually exacerbate the invasion. And so broadly, we have some key questions about pathogen accumulation across all invaders. How long might it take for pathogens to accumulate? And for microspecium, one of the best predictors of whether or not a population would be infected across that big sampling that we did was the time since introduction, with populations that had been around for at least 40 years much more likely to be infected. And then how common, for what species, under what conditions may we expect pathogen accumulation? And then what is the long-term outcome for native species and invaders? And to get at this last question in particular, we've been conducting a series of field experiments trying to put together all the possible interactions between native species, invaders, and pathogens, and how they may interact with each other in terms of resource competition or plant-plant interactions above ground, in addition to interference competition or plant litter interactions where the pathogen is moved through invader litter, which then may have consequences for native species. And so we have really good funding here from the EEID program, which is a joint NSF, USDA, NIH program. And the postdoc on the project, Amy Kende, has been conducting a series of field experiments at that Big Oaks National Wildlife Refuge in Indiana, where she's been manipulating invader and native species density and then manipulating disease load with fungicide and water controls. And she started to put together these models where she can determine under what conditions and what lengths of time will it take for the native and the invader to coexist, or for one to exclude the other. Okay, so now I want to turn to a different study system and to a different potential driver of long-term dynamics and impacts, and that's the role of climate change, and specifically drought. And so when I moved to the University of Florida about 7.5 years ago now, I was struck by these seasonal droughts, which there's a lot of different definitions of drought, but they seemed like droughts to me in the spring and the fall, when we would have, despite the very abundant rain we have in the summer, these very long periods of dry periods of native species tolerating these conditions where it wouldn't rain for perhaps 6 or even 8 weeks at a time. Well, those periods, those dry periods, just dry days in general, are expected to significantly increase across the Southeast, and in particular in Florida. And so I was interested at the same time in this invader, Kogan grass, Imparata cylindrica, which is a perennial rhizomatous C4 invader of woodlands and open habitats and disturbed areas. And in particular, this invader was introduced in both to peninsular Florida and also to Mobile, Alabama, and has spread to a wide range of areas in the Southeast, and it's considered highly problematic. My lab has been working on questions with this invader essentially since I arrived here in Florida. And so the study system that we're interested in terms of impacts is this longleaf pine, Pinus palustris forest or savanna system, and this was the dominant ecosystem type throughout much of the southeastern United States historically. You see that range in purple there, and it's now limited to less than 3% of its historic range due to a combination of factors, including land use change and agriculture, as well as invasive species and other factors. And so historically, we had these very open woodlands that were primarily maintained by wildfires that returned on a 3 to 5 year interval, and now we maintain these systems in terms of species distribution and diversity using prescribed fire. And so we had this general question of how does the interaction between a climate change factor, in this case drought, and invasion by a non-native plant species, in this case Kogan grass, affect a native ecosystem such as longleaf pine forest. And so in the top photo here, you see a really nice, well-maintained longleaf forest here in north central Indiana, or Florida, excuse me. And then in the bottom, you see a similar type of forest that has been invaded by Kogan grass. So the way that we got at this question was with a common garden experiment, which has also now become a rather long-term experiment. This is a simple factorial experiment where we manipulated the drought, simulated drought using these rainout shelters. So we had drought or ambient conditions. Then we had plots that were reference plots or control plots, and then ones where we experimentally introduced Kogan grass. So just to give you a little bit more information about the setup of this experiment, we set up the plots and planted them in the spring of 2012 with native species. We added 20 longleaf seedlings, 12 species of native graminoids and forbs, and then dozens of other species colonized from the seed bank in the surrounding area. 1 year later, after the native species were well established, we added rainout shelters in early spring 2013. You can see the diagram of the shelters there. They have polycarbonate roofing that's light permeable. We have above ground aluminum flashing to prevent overland flow of water, and in-ground barrier to prevent subsurface flow of water. We have a gutter and drain system to take the water off site. 2 months later, we added Kogan grass to the invaded plots from an on-site population. So first, to describe the effectiveness of the treatments, here I'm showing soil moisture over time from 2014 into 2018. And I want you to notice two things. First is that the soil moisture was reduced by 30 to 50% in the drought-treated plots. So our rainout shelters worked in simulating drought conditions, or at least what we thought were drought conditions. But the other thing I want you to notice is that the invasion actually mitigated the effect of drought. And so drought alone is the orange triangle. Drought plus invasion are the red triangles. And so there's actually higher soil moisture in the drought plus invasion than the drought alone plots. At the same time, the invasion worked very well. We added 9 seedlings or ramets of Kogan grass to each plot. Every one of them survived and quickly became abundant in the plots. So we added them in spring 2013. By the beginning of 2015, they were quite abundant, and on average, the invader has been about 40 to 60% cover in the plot. There's a little bit of indication that it suffers from the effect of drought, but it's rather minimal. You will note there that there's a hard freeze that knocked back Kogan grass in the winter of 2018, but I'll show you at the end that it quickly recovered. In terms of the native species effects, here's total resident species cover. I want you to notice 2 things. The first is that there's seasonality in native species, and so they senesce at different points during the year. Obviously the winter. There's a relatively minor effect of the drought, so the orange triangles compared to the light blue circles, but there's a massive effect in terms of resident cover due to the invasion. Notice here that there's a seasonality in resident community cover, whereas we flip back here to the Kogan grass and we see that it does not exhibit the same seasonality, suggesting that part of its competitive mechanism is that it remains green and photosynthesizes throughout the year. In terms of impacts of invasion, we see about 20% less diversity of native resident species under the drought treatment, but we see 60% less diversity under the invasion treatment. So small effect of drought, really big effect of the invader. Now, one of the primary things we were interested in here was the foundation tree species longleaf pine. And so what I'm going to show you here is a series of figures taking us from 2014 to just last year in terms of the survival of longleaf pine, and at the same time show you patterns in soil moisture. So the figure in the far left is the soil moisture in 2013, and then the survival of tree seedlings in the spring of 2014. And to orient you here, the axes are, for the x-axis is flipped maybe a little bit than what you would think about. So it's drought and then ambient rather than the other way around. And that's in order to match the increasing soil moisture that you see in the far right panel. So what we saw, first of all, is that the drought treatment worked. So on average, there's less soil moisture under drought than ambient conditions, but only when they were not invaded. So under the invaded condition, the orange triangles, you see that soil moisture is the same regardless of the drought treatment, again suggesting the invader is mitigating or moderating the effect of drought. Likewise, in response, native tree seedling, longleaf seedling survival match that pattern exactly. So what you see here is that drought is bad for tree seedling survival, not surprising. Invasion is bad for tree seedling survival, but the combination of drought and invasion results in the identical amount of tree seedling survival as neither drought and invasion. And in the right panel, you see that the blue line shows you that with greater soil moisture, there's greater survival of tree seedlings, but this effect goes away when you add the invader because of the moderation of soil moisture conditions. And so the conclusion here from the first year and actually the second year is that the invasion facilitated pine seedling survival by moderating those drought conditions. And so early on, the first couple of years, it was very surprising to us to see this effect. And we thought, well, it looks like we're going to publish a paper that shows that this invader in particular can moderate a climate change factor and contribute to survival of this foundation tree species. Fortunately, we didn't get around to publishing that paper because patterns changed over time. In particular, the invader became much more abundant by 2015, and especially 2016 and 2017. And what we eventually saw is that although the invader still moderated soil moisture conditions, that was no longer the limiting resource. In fact, as the invader became more dense, the limiting resource was light availability. And so tree seedlings that did not make it above the canopy of this invader succumbed to the effect of the invasion. They were no longer facilitated by the moderating effect of the invader on soil moisture conditions. Okay, so another interesting question here, a biotic factor that may drive long-term patterns in these invaded communities is the role of secondary invaders. And so we know now that in many systems, we're not facing one invader, but we're facing multiple invaders. And so I've long been interested in how these multiple invaders might be interacting. And so this is some excellent work done by an honors student, undergraduate student, Emma Byerly. And what she did was to take Chinese tallow, lantana, and Brazilian pepper and experimentally introduce them into these plots under these invasion and drought conditions. And what she saw generally in terms of average biomass of these species is that the invasion, in the case of Chinese tallow, facilitated the species by 238%, so 238% greater biomass due to the invasion. For lantana, we saw this interesting interaction where there's 10 times more lantana biomass in those native-dominated plots because lantana, we think, is much more drought tolerant, and the native species suffered from the effect of drought. And then there's also this positive effect of the invasion overall on lantana. For Brazilian pepper, we see a much more modest increase compared to the others, but 70% increase in biomass of the secondary invader is quite important on its own. So the general take-home from this part here is that although we saw that invasion and drought both suppressed the native species, it facilitated these secondary invaders. Okay, so the last topic here is to think about the role of fire in this system. And so I mentioned early on that natural fires maintain longleaf pine ecosystems, and that we currently use prescribed fires to take the place of those wildfires to maintain longleaf systems that we're trying to restore or maintain. And so we asked this question last year, and what we did was to go in and take down all of those rainout shelters and those control structures, all 40 of them, and then we applied prescribed fires to all 40 plots, and we measured fire intensity and survival, damage and survival of these trees. And so we did this in multiple ways. So you see on the left, passive flame height sensors, which are these T-bars with fire retardant soaked string hanging from them, provides a relative measure of flame height. And then we use thermocouple data loggers placed at ground level, 25 centimeters and 50 centimeters, to measure fire temperatures every second. So first of all, what I want to show you, not surprisingly, is that flame heights were much different in the invaded plots. Regardless of drought treatment, we saw that flames were about 4 times taller. And we, not surprisingly, also saw that the char height was similarly different on these trees. And notice here that the trees are much bigger than I showed you in those photos early on. But at this point, some of these are 3 to 4 meters tall. So we also saw that the fires were much more intense in terms of fire temperatures. And so here I'm showing maximum fire temperature on average under ambient and drought conditions at 0, 25, and 50 centimeters in height. And you see this really strong effect of the invasion where fires are much more intense in invaded plots. And in particular, at 50 centimeters, we see on average over 300 degrees higher maximum fire temperatures due to the invasion. And fires not only burn hotter, they burn hotter longer. So here I'm showing the number of seconds over 100 degrees C. And again, we see a strong effect of the invasion, particularly at 50 centimeters, where fires burn more than 70 seconds longer at more than 100 degrees C. So you would think, of course, that the fuel load would be driving differences. And overall fuel load between reference and invaded plots may be contributing, but in fact, there was not that big of a difference. However, what you see here is that on the x-axis, we have fuel load, the y-axis, maximum fire temperature, and we see a positive relationship. But notice at 25 and especially 50 centimeters, the separation between the two lines indicated that it's not just fuel load on its own, but something else about the fuel that causes this difference. And at 50 centimeters in particular, at any given fuel load, maximum fire temperature was over 300 degrees higher with invasion. And so we think that the fires are more intense due to the differences in fuel structure, not necessarily fuel abundance or not only fuel abundance. So as a result, we looked at, we saw very big differences in terms of damage to the trees. And so here on the y-axis, I'm showing percent of these longleaf tree canopies that were damaged. And the first thing to notice here is that there was twice as much damage in the invaded plots under drought conditions. However, under reference conditions in drought plots, there was 36% less damage. And so there's an interesting interaction here. And then as a result, ultimately what we are interested in is whether or not these trees survive the fire. And so under ambient conditions, regardless of invasion, we see 90% survival of the trees. Under reference conditions without the invasion, but under drought conditions, we see 100% survival of the trees. However, the combination of drought plus invasion resulted in only just over half of the trees surviving. And so the reason why we think this occurred is tree height plays a significant factor in whether or not a longleaf tree can survive fire because of the potential damage to its apical meristem and other factors. And so the tree heights were very different due to both the invasion and the drought treatment. So the left two bars under ambient condition, you see that the invaded trees in invaded conditions were actually taller, presumably to avoid the shading effect of the Kogan grass. However, trees under drought and invaded conditions were not able to grow any larger because of the drought, presumably. And so as a result, this drought invasion interaction resulted in more intense fires, but trees were shorter and therefore greater tree death. So what we're trying to do now is to look more at the longer term picture. So if we think about the grass fire cycle, where a non-native grass such as Kogan grass or Microstesium invades, results in abundance, continuous fine fuels, higher intensity and more widespread fires, and therefore enhanced fire severity for native species, but relatively less damage to native species. So then you can have this positive grass fire cycle. And I have a new PhD student working on this question by continuing to track survival of trees and native species abundance and diversity in the plots. And so here's that data from on Kogan grass, where we see variation in Kogan grass cover over time, over a 5-year period now, where you see that big drop in February 2018 due to that hard freeze. The fires were applied in April of 2018, and then you see this big boost in Kogan grass cover following the fires. Okay, so then to bring us back to this hypotheses table, in terms of biotic and abiotic drivers, we see variable effects or responses in terms of the hypotheses we put forth at the beginning. And, I'm not going to go through each one of these individually, but what I want to point out is that here we have essentially a collection of case studies that informs each of these hypotheses to some degree. And we'd like to have broad models in order to predict when each of these factors and where and for which species, which ones would be important. But at this point, I think we're really at the stage of collecting case studies on longer term dynamics and impacts of invaders. And so then finally, to bring us back to some general comments here, I'll say that these interactions among biotic and abiotic drivers, they may be particularly difficult to predict. We certainly didn't expect that drought plus invasion would actually be, at least initially, good for pine trees, that there would just be offsetting or moderating effect of the drought by the invader. Second, these longer term studies are needed. So if you think back to the Microstesium effects in terms of species diversity, we would have had very different conclusions if we would have looked early versus later in the experiment in terms of what do we conclude about impacts of that invader. And so these longer term, broader scale studies are needed, and eventually we're going to need to tie these into more general models. Third, I would argue that these experiments are needed. So these possible interactions are kind of can be very complicated, and so we need a collection of these case studies. Eventually we're going to need to synthesize them, but at this point I'm aware of very few of these types of experiments that are ongoing. And then finally, I think these types of experiments need to have management considerations, because many of these drivers can be particularly important in different systems, and we need to be able to understand what managers are facing out in the field. And so I think the experiments need to focus on particularly problematic invaders. Here, Microstesium and Kogan grass certainly are of major management focus, instead of focusing simply on species that are the most easy to manipulate or happen to be in nearby proximity to a research university. Okay, thank you very much.
Thank you, Dr. Flory. That was very interesting. I have a couple questions just to start off with. So I noticed that in your microstegium experiment in Florida, it looked like you had tall metal barriers around each of your plot. Was that correct?
In Indiana, you mean?
Oh,
the
sorry.
first,
Yeah.
the microstegium experiment for when we conducted the fire?
Yeah.
Yeah.
So did that have any effect on the microclimate within? So did it get hotter because they were metal barriers inside there? I was just wondering if you investigated if there was any effect of the barriers themselves.
Yeah, I should have pointed out that metal barrier was actually movable. And so it was just held together with bolts and wing nuts, and we would set it up in order to conduct the fire so the fire wouldn't spread to the rest of the plot. So we put it in place. We actually sprayed water around the outside of it so that the fire wouldn't creep under the metal barrier and spread to the rest of the plot. And then we would apply the fire to the inside of the metal barrier and then just remove the barrier just minutes later.
Oh, okay.
And so, yeah, and so it could have actually changed fire behavior a little bit by containing it there in the middle, but it was the best thing that we could come up with in terms of containing the fire.
Oh, okay. So I was thinking they might have been there the whole time to define where your plots were and to keep the invaded areas from not spreading to the uninvaded areas, or okay.
Yeah, so the metal barrier was movable, but there was around each plot we had erosion fencing in order to prevent seed movement. I don't think those affected microclimate conditions in the plot. It could have affected access by rodents, but I actually don't think so, and all plots had it regardless.
Yeah. I was going to ask if there was anything, if you did look into, well, no, actually, my question was more because you were talking about the biological control you were looking at for microstegium, if there's any benefit to increasing seed feeding populations as a maybe native species kind of that you could use as a biological control. Or if there's just so much seed, I don't know.
Yeah, so right, that's really the question. So the biological control, you may notice, or maybe didn't, but I didn't use the word biological control.
Oh, okay.
When I first started finding this pathogen, and other people actually documented this years earlier, but it wasn't very widespread, and there wasn't a lot of attention given to it. There was a lot of talk about whether or not this could be a biocontrol for microstegium. It's almost certainly not possible because it's the same pathogen or very closely related to a major pathogen of corn and turf grass, and it evolves very rapidly. It's not likely a biocontrol. But yes, the question was, will that pathogen suppress enough of the seed production to have population level consequences? So in a given area, say microstegium produces 10,000 seeds, does it matter if it only produces 6,000 seeds that year because it has the pathogen? Those are just made up numbers, but that's what we're trying to figure out now. At what level of reduced reproduction are there implications for suppression of the invasion over time or responsive native species?
Yeah. So let me just remind everybody who's in the webinar, you can add questions to the question pane, and I will read them off to Dr. Flory if anybody has any questions. I'll ask him a couple more questions, and then I can ask any that anybody from the audience puts in there as well. I did like that you had the two different grass species, the Kogon grass and the microstegium, because they are very different, even though they're both invaders. One's, like you're saying, an annual. It reproduces by seed. Kogon grass is a perennial. It mainly reproduces by the rhizomes. I thought that was very interesting. I did notice, the audience may not know this, I am the EDMATS data coordinator, and we did add the Alabama Forestry Commission's Kogon grass data to our database last year. So Alabama is much darker green now from that added data set. So just letting you know, as well as the audience, there is a lot of Kogon grass across the southeast, especially in Florida and Alabama and Mississippi. Um, so, does anybody else have any questions?
Thanks for pointing that out, and I will look at the update of the distribution map for Kogan grass. I've always thought that it was underreported. Um, there's certainly plenty of it around when you drive certain parts of Florida and certainly Alabama.
Yeah, there are certain species that every now and then we come across, like how the maps most certainly don't look like the data that we have. So reaching out to the different people who do research or the different groups who do management, it really helps fill in those different gaps. So yeah, we had gotten the Alabama data last year, so it, like I said, it really filled in a lot for Alabama, and a little bit, I think it did get a little bit into like the panhandle of Florida, because some of those different groups work together across state lines.
Is that the data from the big management effort that they did in maybe 2008, 2009?
No, this is specifically Alabama Forestry Commission data, so it's everything from people who work at the forests and the parks and different things like that, as well as the contractors that they hire. All that data ends up going into the Alabama Forestry Commission database. And because that data is in EdMaps now, it's actually available for use. So you could,
Okay.
if you wanted to do some sort of study with, involving locations, you could use the data from there. It's freely available.
Okay.
One
okay. Other thing,
we have one question. I was gonna, if you don't mind.
So I don't forget, I did want to point out that we added that new module, or I'm not sure what you call it, to EdMaps this year, where we're looking at what pathogens on any invaders. And so if anyone online is familiar or uses EdMaps, and I'm sure there are people, please, if you see pathogens on invaders, please answer those questions when you input data.
Yeah, it's through the seed map, I believe. So if you're out, recreating, going around, if you see an invasive species, if you're reporting it through the seed map, if it's a plant, it'll ask you, do you see damage to this plant, and prompt you for pictures if you answer that yes.
Yeah.
So, and that helps out researchers like Dr. Flory.
Yeah, it can be very helpful.
So Catherine asked, are the 2 grass invaders currently being controlled with herbicides on a broad scale? How are managers currently deciding the control mechanisms today?
Yeah, so first, microspecium is something I worked on in terms of herbicide control, and I was struck early on by the number of people using broad spectrum herbicides such as glyphosate when in fact there are good grass specific graminicides that are effective on it. And so we had a paper that showed that the effectiveness was very high in terms of post-emergent grass-specific herbicides, and that you could get a lot of native species diversity back by using those more specific herbicides. You can also use some pre-emergent herbicides that were extremely effective, but of course that reduces the native species in the area, ability of native species to colonize, because these pre-emergent herbicides are not specific to any functional group. For Kogan grass, we actually have done some work on it. There's a lot of work on herbicides, of course, for Kogan grass because it's such a massive problem. It's actually a federal noxious weed, meaning that I believe state and federal highways have to control it. They have to control it along the highways. You can see that in Florida, where they will control the population right up to the fence line, right in the right of way. And then, of course, it probably just grows back the next year. So there's been, most people use glyphosate and imazapyr in some combination. I don't know if they're using anything else right now. But the question we have is if you can use a grass specific on Kogan grass with any effectiveness, and also in that case have greater native species diversity and cover following removal.
All right, Dexter asked, in the Kogan grass fire study, in the plots with native understory species, was there ever instances of invasion by Kogon grass, probably by seed? If so, any idea if there was a relationship between number of species and the invasion by Kogon grass?
That is a great question. So I didn't mention here is that the two introductions of Kogan grass, one to Mobile, Alabama, and the other to Florida, came from different parts of the native range in Asia. And so the situation is that the Alabama populations, more often, I don't know the proportion of populations, but much more often produce viable seed. In Florida, there's ongoing research on this, but very few, very few populations produce any viable seed. And so, as Rebecca mentioned, the majority of spread of Kogan grass in Florida is by rhizome. And so, in this case, we didn't have viable seed production in our experimental plots, what you see here right after the fire. And so the spread, we didn't have spread into the other plots. That is a great question. It would be really interesting to evaluate. What we do see, and you can see the herbicided area around each plot, is that the Kogan grass spreads by rhizome out of the plot, or tries to, and we have to control it, but we've managed to keep the uninvaded plots uninvaded.
Okay. I didn't know that Alabama had produced viable seed. I had heard only about that wasn't a thing yet because of the being clonal populations. So I didn't.
I know that's the case in Florida, and actually, I should find out more about that. I am not certain. I don't know how much of Alabama, how many of Alabama populations or how much seed is produced, but my understanding is that they do.
Okay.
I don't know where I'm getting that information, actually, now that you asked that or that we were talking about it, but certainly we don't have seed production in Florida.
Yeah.
Not very much.
Okay. So does anybody else have any questions? These were really great questions. So Catherine said, thank you for answering her question. Yeah.
You're welcome.
All right. Well, if we don't have any additional questions, again, I want to mention that this webinar is being recorded. It will be made available. Like I said, I believe it'll be either embedded or linked probably through the NAIPC website. That's NA-IPC, I believe. Let me just double check that. And as well as the Bugwood YouTube channel. So yeah, na-ipc.org. So it should be made available through there. And I want to again thank Dr. Flory for this really great and informative webinar. Probably one of the best ones I've heard in a while. So if anybody, yeah, I think that's about it. All right. So thank you again, Dr. Flory.
Thank you. Thank you for having me. Thanks everyone for listening.